Welcome to the lecture 8. Today we continue our exploration to the physical instrumentation used in spectroscopy. In the previous lecture we discussed light sources. Now we move forward along the optical path. We must answer a practical question. Where do we place the sample? The answer is deceptively simple. We use sample containers, commonly called cuvettes or optical cells. Although they appear simple, cuvettes are actually precision optical components. Their design directly affects measurements accuracy. The most critical factor is a material selection. If you are working in the ultraviolet region, you must use cuvettes made of high purity quartz or fused silica. Why? Because ordinary glass absorbs strongly below approximately 350 nanometers. If glass is used in UV experiments, the light never reaches the sample. Your signal is completely blocked. However, quartz remain transparent down to about 190 nanometer. This makes it ideal for UV spectroscopy. Now, consider the opposite case. If your work is limited to the visible or infrared region, roughly 350 to 2000 nanometers, then silicate glass cuvettes are perfectly acceptable. Even plastic cuvettes can be used. They are inexpensive, disposable, and highly convenient for routine measurements. Let us now discuss the geometry. The globally accepted standard path length is exactly 1 centimeter. This value appears directly in the Beer-Lambert's law. Specialized cuvettes do exist, 0.1 centimeter for concentrated sample and up to 10 centimeter for extremely dilute solutions, but 1 centimeter remains the universal default. You may occasionally encounter cylindrical cells. They are cheaper to manufacture, however, they introduce a serious problem. Alignment sensitivity. If the cell rotates even slightly between blank and sample measurement, reflection angle changes. This produces scattering errors. Therefore, square cuvettes with fixed orientation are strongly preferred. Let us review essential laboratory practices. That is, never touch optical faces with the fingers. Remove grease and fingerprints immediately. Clean before and after every measurement. Never heat and dry cuvettes. Allow natural air for drying only. Periodically calibrate using standard solutions. Always remember, in spectroscopy, small optical imperfections produce large analytical errors. Let us now see how all components combine into a complete instrument. Now that we understood individual components, we must study how they are integrated. Spectrometers are classified according to their optical architecture. There are 4 major designs: single beam spectrometers, double beam in space instruments, double beam in time instruments, and multi-channel systems. Each design represents an engineering compromise between cost, mechanical complexity, stability, and measurement accuracy. Today's lecture focuses on the foundational system, that is the single beam spectrophotometer. Understanding this design is essential because all advanced instruments evolved from it. Let us follow the path of a photon through this system. Imagine a single photon beginning its journey. On the left side, we start with the light source. It produces a broad spectrum of radiation represented as H nu. This light enters the monochromator or optical filter. Here, almost all wavelengths are removed, only a narrow band survives. The beam now becomes approximately monochromatic. Next, the light encounters a shutter. This component is extremely important. The shutter blocks light temporarily so the detector can measure the dark current. Dark current is the detector's intrinsic electronic noise when no photons are present. Measuring this noise allows accurate baseline correction. After the shutter opens, the incident beam with power P0 reaches the sample region. In a single beam instrument, the workflow is manual. First, you insert the reference cell. This establishes 100% transmittance. Then you physically remove the reference and insert the sample cell. The transmitted beam now has power P. The surviving photons reach the photodetector. The detector converts photon into a tiny electrical current. This signal is extremely weak, therefore it passes through an amplifier. Finally, the processed signal reaches the readout system, which calculates absorbance and displays the result. A complete measurement has now occurred. Let us summarize the architecture. A single beam system contains a light source, a wavelength selector, interchangeable sample and reference cells, a detector, and signal processing electronics. But there exists one non-negotiable requirement: voltage stability. Consider the measurement sequence. You measure the blank, you remove it, you insert the sample, several seconds pass. If lamp voltage changes during this time, the incident intensity P0 changes. Your reference measurement no longer matches the sample measurement. The calculated transmittance becomes incorrect. Even a tiny fluctuation can introduce large errors. Therefore, single beam instruments depend critically on highly stabilized power supply. This limitation ultimately motivated the invention of double beam systems. But before moving ahead, let us examine variations of single beam instruments. Single beam instruments exist across a wide spectrum of complexity. At the simplest level, teaching laboratories use basic systems. These may include battery-powered tungsten lamps, colored glass filters, standard test tubes, photovoltaic detectors, analog meters. They are inexpensive, robust, and easy to operate, but precision is limited. At the opposite extreme, we have advanced research systems. These instruments feature full computer control, wavelength coverage from 200 to 1000 nanometers, automatic lamp switching, high resolution grating monochromators, adjustable slit widths, photomultiplier tube detectors, and digital signal processing. Their advantage includes high sensitivity, improved spectral resolution, wide spectral flexibility, seamless software integration. However, and this is crucial, even the most advanced single beam instrument still suffers from one fundamental limitation, that is dependence on source stability over time. To overcome this problem, physicists developed the double beam spectrophotometer, and that will be focus of our next lecture. Today we learn how cuvette materials affect measurements, why path length matters, how spectrometers are classified, the optical path of a single beam system, and the strengths and limitations of these architectures. In the next lecture, we will see how double beam instruments solve the stability problem and dramatically improve the measurement accuracy. Thank you.